Anyway, to be honest, my hands-on session is not very interactive because there are not many intermediate steps. But as long as you have the Docker, we will be able to run it yourself either today or when you want to play at home with it. So let's start then, okay? So now we can say good morning. This talk today is about bridging multiscale in TVB, or about how TVB is trying to make friends with other simulators that simulate the brain at a much finer scale, neural spiking simulators. First, I'd like to motivate a little bit the whole idea. Well, as you know, and I hope you will be persuaded by the end of this workshop, TVB is quite unique as a brain simulator in that it can connect brain anatomical data that we record from individual persons with the neuroimaging data that we also can record from these people. And this is possible only because TVB is a simulator at the large scale of the brain. We simulate sources at a level of description that is responsible for the generation of neuroimaging signals. So this is a unique value of TVB. However, there are cases where neuroscientists really need to simulate networks of spiking neurons in order to investigate specific local systems, neural mechanisms, the function of neurotransmitters, etc. In most cases, when people build spiking networks at that fine scale, the rest of the brain becomes a context for those networks. And usually it is modeled as a Poisson spike train, following the idea that every neural population receives a generally random signal from the rest of the brain. Or when they want to study oscillations, they will usually create some oscillatory, like a sinusoidal signal, to drive the spiking network. However, we believe that there are many cases, as you will see in the following, where these networks, it's meaningful to embed them in the large scale of a simulator like the virtual brain. How would this be possible? Well, the interface between these two levels can happen at the population level. Why is this the case? First of all, because as you saw yesterday with the phase plane models of TVB, usually specific state variables of the mean field models of brain regions correspond to summary descriptions of the dynamics of neural populations. For instance, we can imagine here a mean field model that has two state variables, one representing the activity of an inhibitory population of neurons and the other of a population of excitatory neurons, X1, X2. At the same time, we can average the activity of neurons within a population also at the fine scale. And therefore, this population level can become the interface between the two scales. And this is actually what we're going to do. There are many spiking simulators in the market, like Nest, Neuron. I'm going to show you an interface implemented between the virtual brain and Nest, which is one of the state-of-the-art simulators right now. Now, why to do this? Because, as I said, in most applications, we're going to be interested in the input of the large scale, TVB, to the fine scale, the spiking networks. Because there will be implementations, and we envision such implementations, where we need to drive the spiking networks with an input that is either specific in time or in space or both. Specific in time would be that it has an oscillatory content that is biologically realistic. We know that the brain generates different rhythms that have different functions and drive spiking networks in different ways. Okay? In other cases, we are interested in the spatial context, the fact that each brain region receives a very specific input from other areas of the brain and this is to some degree, as you saw yesterday, determined by the anatomy, but also there is this notion of functional connectivity, right? Connections that are dynamically created or destroyed during the function of the brain. And there are cases where both are important, both the temporal and the scale and the space structure of an input to a spiking network. But there are also cases where we will be interested in a bidirectional coupling between the fine and the large scale, mainly in order to validate the different mean field approximations, not on local statistics this time, but on large scale brain statistics. In other words, we could answer questions like, how does the functional connectivity of the whole brain changes if we substitute the mean field models with spiking models, at least for some regions of the brain? Okay, so I will go through an implementation as a proof of concept at a very early stage of development. And first, we're going to go through the mathematics and the theory and the mechanics of it. And in the next 2 hours, we'll also see how we can implement this through a Jupyter notebook using the specific implementation. So, as I said, this is a package called TVB Multiscale that it's additional to the general TVB package. You can find it in the virtual brain repository of in GitHub. There is a Docker image that you were asked to already pull because we need also to have Nest installed, and this is not as easy. And yeah, and you can run everything also in the HBP Collab, but exactly due to this image with Nest installed, you also have to have a cluster account from CSCS in Switzerland. That's why we could not run today's hands-on session in HBP Collab, but you need really to install this Docker image. So for most of you, I guess it cannot work to use it in the HBP Collab for the moment. So the specific implementation I'm going to show you uses a model for every region of the brain that has one excitatory and one inhibitory population, like the figure I showed you before. Most of the nodes of the brain, most of the regions of the brain are modeled in TVB as mean field nodes. Using a reduced Wong-Wang model, I'm going to show you the equations of this model soon. Then I select randomly, for the example, 2 regions of the brain, the left and right parahippocampal cortices, to substitute the TVB nodes with spiking networks. And for that reason, I'm using a spiking neuron model to build those networks that resembles very much the original Wong-Wang-like spiking neuron model that was used to generate the mean field approximation. Okay? Remember, every mean field approximation of a population approximates the average behavior, or some other statistics as well, of a spiking network. So the 2 models here are in correspondence in the sense that one is a massive approximation of the other. Okay? And as I said, you can find everything also in HBP Collab, but unfortunately you need to have also this account with CSCS cluster. So let's go through the equations of the 2 models so that we understand better the mechanics of the interface between them. This first slide is just to scare you. On the left in green, you have the mean field equations that will govern the dynamics of most of the TVB nodes. On the right, we have the equations of the spiking neuron. So let's go through them step by step. So the state variables of the mean field model are 2. One for the excitatory population, one for the inhibitory population. They are called synaptic gating state variables. Their biological meaning is that they act as a percentage of the channels that are open at all neurons of a population. Okay, so it's a number that goes between 0 and 1. There are slightly different equations for the excitatory population, because here it's the NMDA synapse that governs the dynamics, and simpler for the inhibitory synapse, because in this case it's the GABA synapses, as you're going to see later on, that govern the dynamics. The important here parameter is the rate. There is a rate parameter for both of these equations, one for the excitatory and one for the inhibitory population. This is synaptic gating and rate outgoing from one population towards the others. Okay? How do we calculate this rate? This rate is computed for both excitatory and inhibitory populations by a sigmoidal activation function of the incoming currents. So it's an output rate given by a transfer function that depends on the input currents to every population. And how does this input current is computed? Well, it consists of different parts. If we start with the excitatory current, first of all, there is a constant external input. There is a self-excitation, so current from the same population, excitatory neurons, towards themselves, one to the other, right? There are many neurons, so on the level of the population, it's a self-coupling. Then there is an inhibitory coupling coming from the inhibitory population of the same region. Then there are excitatory couplings coming from all other regions of the brain, from the excitatory populations of all the other regions of the brain. And here you can see the TVB connectivity weights and time delays that are constructed based on the anatomy of the patients, etc. And another current that plays the role of the stimulus in case we want to apply some stimulus, some external stimulus. Going now to the inhibitory population, it also receives a constant external current. It receives excitatory coupling from the excitatory population of the same region. It receives an inhibitory coupling from itself, one inhibitory neuron against the other. And it may or may not receive excitatory coupling from other brain regions. This depends on this parameter that can be between 0 and 1. This is called feed-forward inhibition. For some application in this model, it's needed. In some others, researchers choose to disregard this input. In other words, this is a model that communicates synaptic activity among brain regions. This synaptic activity depends on the postsynaptic rate. This postsynaptic rate depends on the incoming currents. And currents are created by multiplying synaptic activity with the current constant. So synaptic activity directly translates to currents. Okay? This model is an approximation of a spiking network that follows the equations you're going to see now. It is an integrate and fire neuron. That means that the main state variable is the membrane potential, which receives a series of currents from different synapses. And when it reaches a threshold, it will fire a spike, and then it will stay plugged to a specific value, the resting potential, for a specific time, refractory time. During that time, it cannot fire any more spikes. That's the mechanics of this model. And now we need to see the specific currents that drive the dynamics of the membrane potential. First, we have the leak current. We have AMPA synapses. We have NMDA synapses. We have GABA synapses. We have AMPA synapses coming from other brain regions. And for these two terms, for the moment, I will only tell you that they correspond to specific stimulations devices we can create in the nest spiking network in order to stimulate the neuron. I will tell you the exact meaning of them later on, okay? So let's see each one of the currents, how it can be generated. So the leak current is just a conductance times the difference of the membrane potential with the leak potential. Similarly, AMBA synapses follow this equation. Again, there is a synaptic, there is a conductance constant, the difference with the reversal potential of the excitatory reversal potential, and a multiplication with the synaptic gating variable, the percentage of synaptic channels that are open for the AMBA synapse. A similar linear form exists also for the AMBA external synapses that come from different brain regions. Only this time they are weighted by the connectivity weight of the TVB. And similarly for the GABA synapse, which is here. It's also a linear synapse, so it has a conductance, the difference with the inhibitory reversal potential, and the synaptic gating variable for the GABA synapse. NMDA has a more complicated nonlinear expression that you see here. The other inputs I will explain later, as I said. So we have a potential, it receives currents, currents come through synapses. That's the short story. As for the synaptic gating variables, AMBA and GABA have linear differential equations, so they receive spikes as modeled here as delta functions that at specific time points they are 1, at any other point they are 0. So this is how a spike looks, right? And they are just driving the spikes this linear differential equation. The same goes for the synapses, the AMBA synapses that are receiving spikes from different brain regions. And notice that these are as many as the brain regions. So if we have 100 brain regions in our connectome, each neuron, for each region, we have 99 such synapses that come from the rest of the 99 brain regions. Finally, the NMDA synapse is more complicated. It's nonlinear and two-dimensional. Spikes are received at this state variable, which is linear. And this one drives this differential equation of the synaptic gating for the NMDA, which is nonlinear. It has a multiplicative term here between X and S. And actually, it is this synapse, which is slower in time, that enslaves the fast synapses of the excitatory populations. And this is why, as you saw before, the excitatory population, So the excitatory population has a nonlinearity here between the rate and the, so this multiplicative term here is due to the multiplicative term here. Okay? So you see the correspondence between the mean field equations and the spiking neuron equations. So a network of such neurons with all-to-all connectivity between them and for a specific parameter set will be approximated by a mean field equations like this. And the approximation is the better, the more neurons we have in the network, because they cancel out any particularities of the statistics. Okay, how can we interface the two levels of description? I'll show you three different ways, first of all, where TVB could inform the activity of the spiking networks. So TVB activity coming, from other brain regions, how could it drive the spiking network of other brain regions? This is the question we'll pose here. So, first of all, one way would be to try to convert the rate variables here that are outgoing output rates, as we said, to spike trains, and send these spike trains then to the spiking network. So how to do this? At every time step of the simulation, we get the rate of this region. We convert it to a Poisson spike train, which drives the external AMBA synapses of the spiking neurons. So the rate, the spikes generated here will go to every neuron, okay, to every neuron of the spiking network of another brain region. And remember, we said that these synapses are as many as the other brain regions, okay? Then the spikes will activate this synapse, and this synapse will activate here a current that will then drive the potential and generate spikes when it crosses the threshold. Is it understandable? Okay. But we also said that these variables here, when multiplied with a constant, they become a current. So another possibility would be to actually multiply them with this constant and couple them directly through a current. Okay? Since anyway the neuron receives currents, we can send currents from TVB to the neurons. And this is what I show you here. I'm going to show you in more details how we multiply this with a constant. It becomes a current. And then inside NEST, there are devices that can stimulate the neurons. They can generate currents. And we drive those devices with the activity of the synaptic gating variables of TVB. And those devices then send currents to all neurons. I'm going to show you how. Finally, I have to say something. In the previous two scenarios, at every time step, the activity of TVB is transferred to NEST, and NEST is responsible for the time delays and the weights between the TVB regions and the NEST spiking regions. It is NEST that has to handle these peculiarities of the TVB connectome. However, there is a third scenario in which we can take advantage of the fact that anyway TVB computes the coupling with all delays and weights, and therefore we can directly, at every time step, stimulate the neurons with the current they should receive from the whole brain at that time, in which case it's TVB that handles the coupling. And this can happen through an external parameter that all neurons in NEST have, which is made here in order to receive, to stimulate them. I'm going to go through the same information now in a schematic form. So in the first case, we take the excitatory population rate of every TVB region to drive a device inside NEST that can generate spikes. And this device sends these spikes, both to excitatory and inhibitory populations. Well, certainly to excitatory and to inhibitory populations depending on the lambda parameter of the feed-forward inhibition, okay? With the time delay that is defined by the TVB connectivity. We can tell this NEST device, we can program it to store the spikes for some time and send them only with the delay that we require. In this case, we communicate between TVB and NEST via spikes. The second scenario I talked to you about is when we convert this synaptic gating variable, we multiply it with a current constant, we scale it with the number of neurons, it becomes a current. And then this current drives another device, a device in NEST that can generate currents and send them with the corresponding delay and connectivity weight to the spiking networks inside NEST. So this is a coupling between one TVB region and one NEST spiking region. Finally, in the third scenario, TVB is the one that computes the coupling that should be applied to a NEST spiking node at time t, computed through all time delays and connectivity weights that TVB has, and then directly at that point drive a specific external current parameter. After, of course, it's multiplied with the current constant, etc. So there are two ways to do this through currents and one way to do it through spikes. This was the one way, how TVB can drive a spiking network. But as we said, in many implementations, we might be interested also in the other way around. The other way around is not implemented as an input, as a coupling, but as an update. Why is this the case? We want to take advantage of all the things TVB does to a brain. In other words, you know that the TVB can compute large-scale coupling. It can generate neuroimaging signals for the whole brain. So we want also to have these neuroimaging signals, fMRI, EEG, etc., also for the regions that are modeled in NEST. Therefore, we cannot create a hole in the TVB brain. We want the whole brain to be represented in the TVB model. Therefore, we keep inside TVB the regions that are modeled as spiking networks, and at every time step, we need to update them with the activity from the spiking network. So the interface from the spiking network to the TVB is an update. It's not a coupling, okay? Therefore, both on the left and on the right, we have the representation of a NEST node in red. Only here, it is actually the node as a spiking network. Here, it is as it is represented inside TVB. Now, it's quite straightforward how you can update a mean field node from a spiking node. You just need to count the spikes it emits at a specific time interval and compute an instantaneous spike rate. And then this goes and updates the rate parameter of the specific mean field nodes. Now, if you remember the equations, I will go back a bit. Yeah. Well, let's make it bigger. Okay. Now, if you remember, rates are actually... I'm sorry about this. It's a bit slow. Okay, as you remember, rates are computed from incoming currents. Okay, so if you want to update this rate directly from a spiking network, we need only for those nodes that are modeled in NEST to cancel this update, and at every time step update this rate directly from the spiking networks, and then integrate, as we do for all other TVB nodes, the synaptic gating variables. Okay, so that's the mechanism for the bidirectional, the second direction update. Let me see what else I have. Right. For all this to become possible, there are changes we had to do at the simulator of TVB. Now, this is a little bit technical information, but it helps a little bit to understand how the thing works. First of all, until recently, TVB didn't have this notion of variables that change and are recorded at every time step without being state variables. Okay? In other words, TVB would keep in its buffer history, in its history buffer, only those variables that are needed to compute the delayed coupling. Otherwise, it would only compute neuroimaging signals as monitors. You would have to define a monitor in order to record something else. Okay? But for this implementation, we need to have at least the rates recorded and kept at every time step as a separate entirety, both in order to generate spikes, as I showed you to send them to NEST, as well as for the bidirectional coupling to be updated from NEST when we want to couple both ways. So this is an addition that we'll also get in the main part of the simulator of TVB at a later update. In a few months, it will be something, the modeler will be able to define variables in TVB that are intermediate quantities of the model. Okay, they're not state variables, but are used in the equations. Secondly, I had to generate a whole machinery in order to make all these translations between TVB and NEST. because none of the two simulators in principle has an explicit concept of population dynamics. TVB has state variables, that's the closest thing, that summarize the activity of populations. In NEST, neurons are modeled as numbers. So number 1 is neuron 1, number 2, and collection of neurons as tuples, lists, let's say, of neurons. There was no other representation of a neural population. So I had to program everything in between so that I can, as you will see in the second part, so that we can form commands like couple this state variable of TVB to this population of NEST to make it that easy. So there's a whole machinery to do that, which is added to the TVB simulator. Finally, there is a change also that has to be done in the integration loop, what happens at every time step of the simulation. Because just before integration, we need to provide the coupling from TVB to the spiking network. Then we integrate TVB and the spiking network independently. They could even happen on different threads or whatever. This not now, but in principle, logically, could be done completely independently for one time step, right? All I'm showing you here is one TVB time step. Then, if we are interested to update TVB from the spiking network, this is the time to do that. After we have updated TVB state from the spiking network, we need to compute the large-scale coupling, update the stimulus in order to prepare for the next time step. We have to update variables like this, if they exist, variables that are not state variables but are dynamically recorded. Update the history of TVB, etc., generate neuroimaging signals, and then continue for the next time step. Okay? So many of these steps, like this one did not exist until recently in the integration at each time step of TVB. So this is the end of the talk. We could already I could maybe already answer a few questions if there are before we move into showing you an example of how we can build such a complicated network and perform a multiscale simulation. So would you like to make any questions at this point? Yes?
Sadly, yes.
No, that's good. It's not sad.
So you already have a very good integration engine,
yes.
I assume, in TVB. Why not just to add like a spiking neurons and just integrate over them in TVB rather than, because the communication will take time, right?
Very good question. Let's start from the last part of your question. I'm not sure that the communication takes more time. When spiking networks are big, and they can be big as millions, what really takes time is the integration of the spiking network. TVB just sits and waits, okay? So this is not where the time problem arises from. Why wouldn't we do spiking simulations in TVB? Actually, the last few weeks, I implemented a spiking simulation in TVB. The reason was because I wanted to have a second implementation to validate against this one. However, remember the following. At every time step, TVB will compute, first of all state variables, and second, a coupling among all of them, which is going to be applied to the next time step. So TVB is meant to simulate the whole brain as a unique system of differential equations that you can even study analytically. And these differential equations communicate through continuous variables, which means that there is communication all between all at every time point. Do that for millions or billions of state variables. You need a large amount of memory and a lot of computation time. Instead, spiking simulations simulate every neuron on its own, and only occasionally, when there is a spike, will communicate something one to the other. There are specific protocols for this communication to optimize it in terms of memory and speed. Okay? That's why spiking simulators are completely different. They have a completely different structure. For instance, in Nest, every neural model has inside its definition, also the integration loop. In other words, you can build a network in Nest where one neuron is integrated with Runge-Kutta, for instance, and another one it's explicitly solved, or whatever. It stops being a unique dynamical system that you can study analytically, you can make theoretical assumptions. The assumptions become statistical at that point. So there is a completely different orientation between large-scale simulators, large-scale simulator, because I think it's unique, TVB, and all other spiking simulators. And the role of this project is really to use the strong points of both in applications that we need to do that.
Thank you.
You're welcome. I should have repeated the question though, but I forgot it. The question was why we don't do spiking simulations with TVB only.
Okay, I think you did say that.
Yeah, okay, good.
Yeah.
Any more questions?